Okay. So, welcome everyone. My name is Sharon Rounds. I'm the program director for Advance Rhode Island Clinical Translational Research Center here at Brown University, which includes our partners at the hospital affiliates, the Rhode Island Quality Institute, and the University of Rhode Island. And it's our great pleasure to have a monthly research seminar. And today we're very pleased to have Jake Kurtis, who is the chair of pathology and laboratory medicine at Brown, and also director of the MD PhD training program at Brown. Jake will be telling us about his work on malaria, and by way of background, he's almost a Brown lifer, having been I think you were a PLME student, weren't you?
In the first class. It's frightening.
Wow. And he went on to get both his MD and his PhD from Brown. But he then went off to Africa to complete postdoctoral training in malaria immunoepidemiology, and then to the hospital of the University of Pennsylvania for residency in clinical pathology and fellowship training in transfusion medicine and coagulation. So Jake will be telling us today about his work developing novel drugs for severe malaria. And I would ask if people could put their questions in the chat, and we'll address them at the end of the presentation. So take it away, Jake.
Awesome. Thanks so much, Sharon. And thanks to your whole team who invited me and made this really easy to participate. Really excited. I'm fired up to tell you not about vaccines today, but rather about drugs. Yeah, in so many times, I don't know if this has happened to you folks, so many times in my life, I've been told that I'm not qualified to do X, Y, and Z. So I figured I'd add another thing that I'm not qualified to do, and that is develop drugs. The whole idea here, and if there's any secret sauce in this presentation, it's that one can use, or perhaps one, that's the conjecture, can one use vaccine targets and repurpose them as drug targets? And that's basically the whole story I'm going to try and tell you today. The first part of the talk is really, zeroing in on why we picked a particular vaccine candidate as a drug target. I'm going to try and go through that part of the story fairly quickly. It's all been published. But then we're going to talk about, can we drug this target? And that's where I very quickly get out of my depth. But despite that, I think, we've amassed a great team, and hopefully we've made some progress. So fired up to tell you about it. I am going to be presenting confidentiality. Yeah, I will be presenting some unpublished results at the very end. I just found out it was getting recorded. And so you'll see some very discrete redactions that happen in the last 4 or 5 seconds before going live. I'll be very curious to see if anyone can see what's been redacted. And then lastly, it's funny. There we go. I finally have some disclosures. Sadly, they've done nothing but cost me money. It's kind of frustrating. But I am a founder of a company called Elkert and a company called Ocean Biomedical. All right, so first part of the story, why malaria and how did we make a vaccine candidate for it? So malaria is the greatest single agent killer of children on the planet. Nothing comes close. Currently, there are 3 billion individuals who live in malarious endemic areas. There were 300 to 400 million infections last year. Gives you kind of the sense of the market size for an effective drug. The 3 billion is a sense of the market size for an effective vaccine. There were 627,000 deaths last year, the majority of them in sub-Saharan Africa and the majority of them in children under the age of 5. So it really is a phenomenal killing machine. Jeffrey Sachs, the health economist, coined the phrase where malaria has prospered, people have not. This is a bit of a map of the malarious world. It's also a map of pick the drug, and this is the map of resistance to it. The chloroquine resistance map really flows right over this. No surprise, you know, when I think of sort of development of nations and of peoples, it's no surprise that if you are losing a quarter of your children under the age of 5 in some of these communities, you're really not going to have the economic prosperity and political stability that you would hope for a region. So it really is super important. Boy, did the medical students hate when I used to teach this to them. You know, you're going into battle with an adversary that has kicked your butt for tens of thousands of years. You really do need to know the sort of ins and outs of its behavior, in this case, the life cycle, if you're going to actually try to interdict it. So real quick, female Anopheline mosquito bites you. First thing she does is she spits down her straw to anticoagulate it. And my day job as a coagulation doc, you know, one of the more recent anticoagulants we use, leparudin, actually came from leech spit. People are working on mosquito spit as a source of anticoagulants, but she spits in it so that her straw doesn't, her proboscis doesn't clog. Parasite, it's just crazily adapted. It's like honed in the fiery crucible of evolution. It is sitting in the salivary glands, waiting to go in with the loogie. So in she goes. She spits about 20 or 30 of these things called sporozoites. And in a feat of remarkable receptor-mediated homing, they're cleared from the circulation in 5 to 30 minutes. They infect liver cells. It's typically a 2 to 3 week cycle in the liver. I'm only talking about falciparum. There are 4 or 5 species of malaria that infect humans. 95 to 98% of all the killing is done by falciparum. So I'm really focusing in on falciparum. Sporozoites go in, infect the hepatocyte, asexual reproduction. One sporozoid enters one hepatocyte. About 2-3 weeks later, sometimes shorter, sometimes longer, out pop 10,000 merozoites. So average infection, you get about 30 injected, 15 make it to the liver, 150,000 pop out. These are called merozoites. They infect red blood cells. There's no real liver disease, by the way. Really none, because so few hepatocytes get infected. All the disease happens in the blood cycle. And so what you see here are merozoites infecting red blood cells. They infect incredibly quickly, microseconds. They're outside a red blood cell. So if you're going to stop it at that stage, you got to be fast. Infects a red blood cell, goes through something called a ring stage. I don't know if you can see my pointer down here. Goes through a ring stage, then a amoeboid form that I'm not showing you called a trophozoite, then a schizon, segments into baby merozoites, 16 to 24 of them, then they rupture. So this cycle is about a 48-hour cycle. The candidate I'm going to tell you about is down at the amoeboid form at the bottom of this circle. It's a candidate called GARP. We'll get to it in a second. Asexual reproduction, lots and lots of multiplication, lots of disease. Every once in a while, the asexual reproduction gets boring for the parasites. They differentiate into male and female gametocytes. They get taken up by the next mosquito. So romantic in the belly of beast, they conjoin, form an ookinete, develop in the gut of the mosquito, crawl their way to the salivary glands, and that's how you get transmission. So for those of you who don't look at blood films, this is what a blood film looks like. These are red blood cells up here. These are infected red blood cells with parasites in them. There's occasionally a sinister looking parasite. But if you look at this, this is 50% of these red cells are infected. 24 hours from now, 50% of those red cells are going to lyse. So massive hemolytic anemias can occur with unchecked falciparum malaria. That's one way it can kill you. The other way it can kill you is the parasite wants to avoid the spleen. And to avoid the spleen, it puts on the surface of these infected red cells adhesion molecules that bind to vascular endothelial molecules. It's like Velcro, and gives you diseases like this. This is cerebral malaria. This is brain. This is a capillary. These are not nucleated red blood cells, though they might look like that. They are parasite-infected red blood cells adhering to the capillary endothelial wall, causing ischemia, as well as immune activation And what you see out here is dead, dying, vacuolated brain. So really bad disease. Another disease that occurs, sorry guys, I don't know how to turn this thing off. Another disease that occurs due to adhesion is pregnancy malaria. This is a placenta. This is a villus in here, the figure of 8 looking thing. This is maternal blood, which bathes the placenta. The red blood cells in here on the inside of the figure of 8 are the fetal red blood cells. You'll notice all these nucleated reds, they're not nucleated reds. Those are infected red blood cells. Some of them are adhering. The molecular mechanism of that has been worked out actually by my postdoctoral advisor. It was a beautiful science paper. Notice the fetal red blood cells are not infected. So it really is not it's not typically a vertical transmitted disease, but causes horrific birth complications, prematurity, and significant maternal anemia and hemorrhage. So real bad all around. How do we control it? We have bed nets. Bed nets actually work. Paper here from an illustrious colleague of mine, Jennifer Friedman, one of the first sections of papers to really look at the impact of bed nets on children and school-age children in particular. They reduce mortality by about 17%, which is awesome, but it's not good enough. We still have a lot more mortality we got to deal with. We have insecticides, we have antimalarial drugs. We don't yet have a highly effective antimalarial vaccine. So what's the issue with vaccines? For the most part, vaccine development programs have really been restricted to identifying their candidates in rodent models. And the real problem is mice lie. They don't get human malaria. They get mouse malaria, and mouse malaria is not very similar. So the generalizability to humans has been kind of stinky. We take a really different approach. We start in the field. We follow large cohorts of children over time, typically from birth to about the age of 4, collecting dozens and dozens and dozens of blood films. So we have a really nuanced data set of parasitemia, but we also have a nuanced data set for all the epidemiologic variables that impact resistance. For instance, if you have a higher socioeconomic status, perhaps you have window curtains, or you have a higher quality roof. And so you have fewer mosquito breeding sites near you, live up on the hill, not down by the lake, 1,000 things like that. There are genetic variables. Sickle trait, of course, is a common one, but there are others. So you really have to eliminate all of those non-actionable variables if you want to hone in on resistance. So we identify folks who are either resistant or susceptible to infection. We take their sera, and both in our field laboratories as well as laboratories back here at Brown, we do some DNA gymnastics. We've got a method that allows us to identify parasite genes that encode parasite proteins that are only recognized by the resistant folks, but not the susceptible folks. Then we always go back to the field and we ask the question, if kids made antibodies to those candidates, were they protected over time? Ultimately, we do some animal model work, which I'll show you, but I'm really more of a fan of humankind and really think the immunoepi is a pathway forward. So just to really make it concrete, parasite has lots of proteins on its surface. And susceptible children, I'm showing you an antibody from a susceptible child over here on the left. They make antibodies. Susceptible kids make almost the same antibody repertoire as your resistant kid. What we're looking for are these golden triangles. The problem is there's a sea of blue boxes. And what we need is an approach that allows us to identify the Boolean difference between the antibody repertoire of a resistant child compared to the antibody repertoire of a susceptible child. You might think they're really distinct. I don't know if you guys can see me on this video or just the slides. I'm holding up my hands in 2 circles. The resistant folks and the susceptible folks, their antibody repertoires are not distinct like this. They overlap. So you're really looking for the needle in the haystack. So this study was conducted in Muheza, Tanzania, an area that has an entomologic inoculation rate that is outrageous. Over 300 infectious bites per person per year, almost an infectious bite per day. You know, 1 mosquito bite's enough to ruin the picnic. but one infectious mosquito bite is enough to ruin your life. So we followed almost 1,000 kids when the study was all done, actually just over 1,000. We followed them from birth through 4 years of life. In the first year, we were collecting blood films every 2 weeks. And in the second, third, and fourth year of life, we were collecting them every month. We had active surveillance for sick and hospitalized children. I'll tell you about that in a second. For this particular study, we had almost 50 blood films on every child. And so there really was a very nuanced assessment of whether they were resistant or susceptible. Active surveillance, what did that mean? It meant that we had a health wagon staffed with what would be the equivalent of here of they're a physician assistant or nurse practitioner. They're called clinical officers, with high quality anti-malarials and diagnostic methods. And despite that, the mortality rate in this community of 1,000 children was about 7%, which sounds horrific, and it is horrific. But compared to the 25%, which was occurring in the communities that were not participating in the study, it was actually pretty impressive. So from all this data, we cherry-picked the most resistant and the most susceptible group of kids. 14 really susceptible kids, 12 really resistant kids. We matched them for a bunch of things like hemoglobin phenotype, sex, yada, yada, yada. Here's the important part. At the age of 2, both the resistant kids and the susceptible kids, second line from the bottom on this chart, had lots of parasites. Up until the age of 2, they had lots of parasites. After the age of 2, something really different happened to these resistant kids. They demonstrated their resistance. After having enough exposure, they demonstrated resistance, whereas the susceptible kids went on and continued to be susceptible. So it's really nice here, the power of a longitudinal study. We know that our resistant kids were being exposed, and yet something, they acquired something. And that something, we hypothesize, was an antibody to a unique malarial antigen. So this is just a slide of the DNA gymnastics. It's pretty old school. We've done this both with standard lambda phage, where you screen a cDNA library, you pull 2 filters where you express the protein, you probe one with resistant sera, one with a susceptible sera, and what you're looking for are the red spots that are only recognized by resistant. You don't want the blue dots, which are recognized by both resistant and susceptible. This is sort of what a filter looks like. And what you can see, 1, 2, 3, 1, 2, 3, there's a 4th up here that's missing. That's only recognized by the resistant. So that would be a vaccine candidate. We're actually doing this in a much more sophisticated way these days, using a differential biopanning, where the phage actually have the protein, see it on the coat there, as well as the gene in one particle. So if you biopan on the protein, you get the gene. The key is find the phage that are recognized by resistant but not susceptible. That's the differential piece. So we did this, and we found a protein called GARP, which I like to think of as the donkey I hope to ride to fame one day. So GARP, glutamic acid-rich protein, is we're able to express it and purify it in E. coli, and we're able to express and purify mammalian systems. The piece we cloned was called GARP-A. The full-length protein is shown here. It came up many, many, many times in our screen. GARP's got some funny properties. It's up on the exofacial surface of the infected red cell at the trophozoid stage. I'm showing you that in green in these immunohistochemistry slides. So it's up on the surface of the infected red cell. That's a funny place for it to be. At this point in time, if you look at that green circle, the parasite's about 1/3 of the size of that red cell. So it's pushing this protein across the parasite membrane, the vacuole the parasite sits into, across the cytoplasm of the red cell, across the red cell membrane, and up on the exofacial surface, which is what I'm showing you in that transmission electron micrograph right there. I'm showing you those little dots. That's GARP out on the outer surface. It's on the surface, has 0 homology to any human protein, has 0 homology to any protein in any data bank anywhere. It's a one-off in nature, only in falciparum, and the falciparum variant that infects great apes. No other malaria has it. So it's a real weirdo protein. It has almost no sequence variation across 6,000 parasites that have been sequenced. So on the surface, no homology, so you don't have to worry about immunotox as much if you're going to target it, and very little sequence variation. So I'm not so worried about escape mutants already existing. So that's that story. Operant question. resistant kids make this antibody. Susceptible kids don't make antibodies to GARP. Is the antibody the reason the kids are resistant? Do antibodies against GARP kill parasites? And so sure enough, they do. This is a parasite growth assay on the left, vertical axis parasite growth, horizontal axis showing you different parasite strains. We've done it with hundreds of strains, including freshly isolated parasites. The story is always the same. Treat them with no serum in blue, parasites grow great. Treat them with serum from a pre-immune from a mouse in red, parasites grow great. If you vaccinate the mouse with GARP as a recombinant protein, GARP is a DNA vaccine construct, or most recently, GARP is a lipid-encapsulated mRNA. What you get are antisera in green triangles that essentially kill all these parasites. We've been able to make monoclonal antibodies like 7899 on the far right that recapitulate the phenotype of this vaccine sera. So antibodies against GARP appear to kill parasites. What's weird is how the parasites die. So bottom left, I'm just showing you some field collected. These are fresh isolates from our Tanzanian field site. You see the same thing. Check out these photographs. So pre-immune in the middle upper panel at 24 hours, those are healthy looking trophozoites. And if you follow them down to 48 hours, they've ruptured out, segmented, ruptured out, and reinvaded, and you've got beautiful looking rings. Pre-immune looks exactly like you would expect. If you treat with anti-GARP, already at 48 hours, they're looking a little dysmorphic if you're used to looking at parasites in culture, but by 48 hours, they're shrunken and pycnotic. This is sort of pathognomonic for a process called apoptosis. And so we have a hypothesis that antibodies to GARP trigger apoptosis. They're doing this in the absence of complement and in the absence of immune effectors, immune effector cells. So it's really cognition alone is causing this phenotype. This is just a bunch of data to show you that the parasites really are undergoing apoptosis. On the left is JC1 staining. What that is a mitochondrial, it's a measure of mitochondrial depolarization. And what you can see is by 24 hours in the anti-GARP sera compared to the pre-immune sera, you've basically depolarized all of your mitochondria. On the far right is apostat staining. Apostat is a measure of caspase activation. Technically, malaria parasites don't have caspases; they have metacaspases. But as it turns out, the sequence that is critical in the caspase is conserved. And what you can see is that the parasites treated with anti-GARP are in fact undergoing caspase activation and tunnel staining for DNA fragmentation. They're undergoing DNA fragmentation. So it's looking like apoptosis. Calcium release is another hallmark. The parasite stores calcium in this funny vacuole. So here in this pre-immune TEM image on the top middle, what I'm showing you is an infected red cell. That's the parasite inside. And this vacuole and those crystals, that's the food vacuole. It's both its stomach and its excretory organ. And so all those crystals are heme getting detoxified and polymerized into hemozoin. That's what it's supposed to look like. If you treat with anti-GARP, well, two things. I can't find the food vacuole itself. It's gone. And the hemozoin crystals are scattered. The hemozoin crystals really need to be sequestered. You know, heme and even the hemozoin are in fact under significant oxidative stress. So that's a really funny finding. We really wanted to, we wanted to drill down on that. Turns out there's a calcium dye, fluo4-AM, which in the pre-immune condition is labeling the food vacuole. If you treat with chloroquine, which is a known antimalarial known to disrupt the food vacuole, the dye leaks out. That's our positive control. If you treat with anti-GARP, bottom middle picture, again, the dye leaks out, showing you that calcium is getting released due to lack of food vacuole integrity. And then lastly, here's a serial block face scanning electron micrograph of parasites treated with pre-immune on the left or anti-GARP on the right. And what you can see, I hope this is coming across Zoom, is highlighted in yellow. That's the hemozoin. Notice in the anti-GARP, it's scattered. Then I'm going to show you the food vacuole. It seems to be gone in the anti-GARP treated. Then I'm going to show you the parasite membrane, which is intact in both cases, and then the RBC membrane. And as it spins around, you really can see that the hemozoin is unbounded by any vacuolar membrane to protect it. And so that is sort of a hallmark of parasite death. We always go back to the field. And in this case, we asked, if we measure anti-GARP antibodies in all 700 of those kids, do they predict anything? And it turns out, if you had detectable anti-GARP, you had half the cases of severe mortality. Severe malaria, I misspoke, severe malaria. All mortality is severe last time I checked. You have half the cases of severe malaria, all cause mortality about the same. So that's, getting run over by trucks, meningitis, and malaria. But if you look at malaria attributed mortality, it's about half. We don't hit significance because we're doing our damnedest to reduce all-cause mortality in the cohort. So we're pretty excited. We like to generalize to independent cohorts. In the middle in B, this is a cohort of 12 to 35-year-old males who had blood films every week through a high transmission season. And what you can see is those kids and young adults who had detectable anti-GARP had half the parasites on all those blood films over 16 weeks. That was in Kenya. This was Tanzania, spanning 20 years, by the way. I collected the B. I was part of the collection as part of my postdoc. And then on the far right, this is an ongoing cohort in Kenya of 2 to 7-year-olds. And what you can see, again, whether you look at all kids or kids who got infected, individuals with high levels of anti-GARP had lower parasite densities. So 3 different cohorts we've seen this. We have taken this into non-human primate studies. Remember I told you mice lie a lot. There's a monkey. It's an Aotus monkey. Don't think gorilla, think more, hamster. It's about 1 kilogram. It's a cute little animal. Aotus. They get falciparum malaria. And if we vaccinate them with recombinant GARP protein on the left, or with mRNA encapsulated, lipid encapsulated mRNA on the right, you generate antibodies which control real life falciparum infection that would kill any of us. In fact, the majority of the monkeys self-cure. So that's the vaccine story. The things that I highlighted are the very same things that compelled us to move into trying to drug GARP. No homology on the surface, limited sequence variation. I didn't tell you this. Our monoclonal kills. You can make an FAB, and I never get the primes and the ABs and the parentheses right, but the kind that's monovalent, recombinant monoclonal kills. So you really are not even needing cross-linking, which is kind of important if you're thinking about drugging it. And what I haven't shown you is that GARP appears to be essential for in vivo growth, not in vitro. We can grow a knockout in the Petri dish. We can't get it to cycle in an animal. So with that as the précis, we think that small molecules which mimic the binding of anti-GARP to its target, i.e., they would inhibit the binding of anti-GARP to its target, that population of small molecules should be enriched for drugs that bind GARP and induce parasite apoptosis. That's the conceptual idea. There is one example I was able to find, and if anyone knows of other examples, please let me know. The one example is in flu, where someone took a therapeutic anti-flu antibody and tried to do small molecule drug screening. So we screened 6,400 compounds from the Cambridge Library of small molecule, drug-like small molecules, using this assay. So GARP is coupled to a bead and mixed with a mixture of 80 compounds. So we had 80 mixtures of 80 compounds each for 6,400. And in this mixture, I'm showing you there's a lunar lander that's going to be important. It's going to bind GARP. And in the mixture on the right, there's no active compound. So you mix, you wash, you come in with anti-GARP. If the lunar lander is blocking the antibody's binding site, you'll get less fluorescence because the antibody's tagged. So what we're looking for is inhibition of antibody binding. We did this, and we got 5 mixtures that appeared to have activity, and we've drilled down on the E7 mixture. We screened 10 mixtures of 8 mixtures of 10 compounds, found the hit, and then when we were down to 10 compounds, we took all 10 into a parasite growth assay. And this is what we got. Parasite growth measured spectrophotometrically on the vertical axis, drug concentration on the horizontal axis. I actually got the joy of teaching myself chem draw to draw the molecule. So I had some fun with that. It took hours. In black, throughout this, I'm going to show you what I call the backbone, and in red, the variable group. This is the molecule that we got. And we got an IC50 for parasite killing of about 100 micromolar, which was a nice initial hit, but it's not gonna, it's not active enough to go prime time. I went to go order more of this compound 816 on the website for Cambridge, and buried at the bottom, obscurely, there's a checkbox. Would you like to order the 2- and 3-dimensional analogs? I'm like, yeah. It's a process called SAR, structure activity relationship, SAR by catalog. And so I did that. And for $11 each, I got 48 additional compounds, all with the same backbone in black. We screened all 48 in parasite killing assays, and we improved by 1.5 log our killing activity simply by changing out the methyl for this structure. So this drug called 579, you can, I'll tell you in a sec, you can order analogs of this. But the first thing we wanted to do was look at specificity. So remember I told you we have a GARP knockout parasite that grows in culture, not in vivo. We don't have activity against the knockout. So that is showing you a couple of things. It's showing you specificity for GARP. It's also showing you that this isn't chemotherapy. Remember, a red cell has very few ways to scream in anguish. And so typically, if you do a phenotypic screen for anti-malarial drugs, 99.9% of them would be great chemotherapeutic agents for cancer. They'll kill everything in their path. So we were really excited that 579 did not kill GARP knockout parasites, which have mitochondria, ribosomes, DNA polymerases. So we're pretty thrilled. When we looked under the microscope, this is the knockout treated with 579, and what you see are perfectly healthy, that's a dual infected red cell, perfectly healthy trophozoites. If you treat wild type parasites with the vehicle alone, again, perfectly healthy trophozoites. But if you treat wild type parasites with the drug, shrunken, pycnotic, dying, really dead parasites. Go to order 579, 2- and 3-dimensional analogs of 579 gives you a few additional hits. We tested these, and lo and behold, found another compound that had similar activity. Not going to go through this in fine detail, but suffice it to say, the drug kills by activating apoptosis, as evidenced by mitochondrial membrane depolarization, tunnel staining, and caspase activation. We've looked at 579 on a variety of mammalian cells, BWOs. It's a choriocarcinoma, human choriocarcinoma cell line. No toxicity up to about 100 micromolar. The IC50 for killing parasites is between 5 and 15 micromolar. So, a reasonable selectivity index. It shows no toxicity up to 400 micromolar in human white blood cells, as assessed by an Xn5 and 7 AAD for necrosis and apoptosis. Collaborating, this, by the way, I'll get into the acknowledgements. Just an entire village of scientists have contributed to this. This work was done in our structural core by Mandar Naik and his team. This is surface plasmon resonance, where we demonstrated that both 424 and 579 bind to PF-GARP specifically with a KD of about 25 to 30 nanomolar. This is 2-dimensional NMR. It's called an HSQC experiment, where Mandar took GARP, he labeled it with nitrogen 15, and collected a spectrum, and that spectrum is shown in red. Then he collected the spectrum after incubating the GARP with our drug. This time it was 424. And what you can see, the arrows help me figure this out. In blue are where you've had a spectral peak shift. And that spectral peak shift is evidence of direct drug binding to GARP. We wanted to do some PK, and this is an area I think that we're hoping to improve the enterprise-wide access to PK with a COBRA that Jen Friedman and I are putting together. It was hard to do this because we didn't have anyone who routinely would measure unknown small molecule drugs in plasma. And so I reached out, I think it was Ed who put me in touch to Kurt Pinnell over in engineering. And he said, oh, we never do that, but it's such a cool idea. Let's try. He was awesome. He had all the great instrumentation to do it, and really did fantastic work for us. He and his team. He had a whole team working on this. So what he did, oops, sorry. There we go. He developed an LC mass spec assay for both of our drugs, showed great linearity and sensitivity. And then we did single dose PK, where we gave IP, the drug, at 50 and 100 milligrams per kilogram. We have a desired trough level, which is 2 to 3-fold above the IC50. And we were looking for a dose, route, and interval that would allow us to keep the plasma concentration above that trough level for 4 days. That was the goal. There's a mouse model I'm going to tell you about in a second that's fairly well established. But you typically treat that mouse for 4 days, and we wanted it to be above that trough level. And so what you can see is at 100 milligrams per kilogram, which is a high dose, by the way. But if you actually have ever dosed someone with like piperacillin, you know you have to bring a wheelbarrow full of drug to the patient's room. It's not totally cray-cray, but it is high. We can do that Q8 hours if we give 100 milligrams per kilogram. So we were pretty excited about that. It has a half-life of 2 to 2.5 hours. Just to show you that these things are kind of drug-like, Swiss ADME is an online server for both compounds, and it looks at drug-like properties, the rules of Lipinski and Gose and a couple of others. These are, rules of thumb. Is it less than 400 Daltons? Does it have the right number of rotatable bonds? Does it have the right planar structure, the right lipophilicity? Those are what goes into these drug likeness rules. And what you can see is in the spider web here, that these compounds really are kind of drug-like. So there is a model. Remember I told you we sometimes go to the animal models for malaria? This model is a funny model. This is, it's almost like it's a furry test tube. This is a profoundly immunodeficient mouse. It's the NSG mouse, so it's NOD-SCID gamma. If you transfuse it intraperitoneally with 1 ml of human red cells every day for about 7-8 days, you will replace the circulating erythron of the mouse with human red blood cells. Then you can actually infect it with real-life falciparum malaria. Now, we had to do falciparum malaria because remember, GARP doesn't exist in the mouse malarias. So we did that. And we, in fact, were able to infect these mice with falciparum malaria. And if you look at the vehicle control mice, they went up to 10% parasitemia. It was pretty phenomenal. If you treat with our drug, 579, and the drug treatment was days 3, 4, 5, and 6, and this is the standard, you see a rapid reduction to 0 of the parasites. We were pretty fired up. In the lower right inset there, I'm showing you published, this is not our work. This is showing, this is the most effective drug we have today, the artesunate compounds. The reason they, the discoverer of which won the Nobel Prize about 7-8 years ago for this discovery. The reason artesunates and the artemisinin family of drugs are so important is they are what's called a rapid blood schizonticide. They have very, very rapid, compared to other antimalarials, killing activity. And so what you see is in the published data, they're getting killing over about 3 to 4 days. What really excites me is in the same animal model, we're getting that rapid reduction in parasitemia. So we would love to push these drugs forward. Some of the criticisms we've gotten is, oh, it's got such a high IC50. Now, fair enough. The issue, though, you got to remember, and I've struggled, and I'll be honest, I've failed to convince reviewers at NIH, at least. We did get some great Falk support for this, but I've failed to convince reviewers that it is not the IC50 that matters, but rather the selectivity index. If your therapeutic window is narrow, it doesn't matter where you are on the absolute scale. If you go a little bit above it, you're going to be in trouble. In this case, the drug level we need is relatively high compared to most pharmaceuticals, but our selectivity index seems fantastic. In any event, I absolutely do not think that screening 6,400 compounds is an exhaustive search of the chemical space. And so we moved into another library. We could have just screened more Cambridge, but this library, and see if you can spot the portions that are redacted. This library has some really interesting properties. Where'd it go? There we go. So this library is called the CTS FIU. It is a combinatorial library. I've written about this thing a zillion times, and only recently do I think I really understand how this works. It is a library of 30 million compounds arrayed on a single 96 well plate. Each well in the 96 well plate has a backbone, which is what I'm covering up with the Homer Simpson dough there. Each well has a backbone, and the backbone has between 3 and 5 potential R groups. See that R3 that I didn't cover up with the Homer? There are 3 to 5 R groups on each backbone, and each well in the 96 well plate has a single backbone. If you can have 42 potential substitutions at the R1 site, which is down here, and 26 at the R2 site, and 42 up at the R3 site, if you do the math, that's 45,000 compounds. Once you add another R or another 2 R groups, now you're in the hundreds and hundreds of thousands of compounds, millions of compounds, and that's how you get to 30 million on this plate. You screen the plate using the exact same antibody inhibition assay. We did that. 2 of the 96 wells showed antibody inhibition. This one is the one I'm going to tell you about. It was in the well called 2353. Once you get a hit, then you screen the sub-libraries in which each sub-library has one of the R groups fixed and the other R groups vary. And so just to be concrete, in sub-library 1, there are 42 mixtures. Each mixture has a different R1 that's fixed, and the R2 and the R3 vary. So you have 42 sub-library 1s, 26 sub-library 2s, and 42 sub-library 3s. It's 110 sub-libraries you have to screen. So essentially, it's another 96 well plate and a little bit more. You screen that. Once you've done that, you've identified which R1s, there were 3 of them, which R2s, there were 3 of them, and which R3s, there were 4 of them, have activity. You don't know which combination of them have activity. So now you have to chemically synthesize, it turns out, 36 individual molecules. So we synthesized 36 of these individual molecules. Work done in Florida at our medicinal chemists lab who designed this library. It's a very amazing library. And this is what we found when we looked at those 36 compounds. A bunch of them had significant activity in the nanomolar range. This is 1,000-fold more potent than 579 and 424. So this is a parasite growth assay on the left, and I'm showing you 3 compounds. We're trying to winnow down from 36 to a more manageable number. These are our favorite 3 at the moment, 17, 19, and 20. Reproducibly kill 3D7 parasites. I'm not going to show you the data, but reproducibly kill parasites that are drug resistant to chloroquine, quinine, and pyrimethamine. Reproducibly kill parasites that are resistant to artemisinin, and reproducibly kill parasites that have been recently isolated from the field. I am showing you DD2. So this is the chloroquine and pyrimethamine and quinine resistant parasite at the same IC50. We've looked at multiple measures of cytotoxicity, including in E. coli. This was actually done by the high school students who rotated in our lab this summer from the Path and Lab Medicine's high school outreach program. So those high school students, hopefully one day soon, will get published. 75, 76 micromolar. That's more than 1,000-fold selectivity. This is a tumor cell line, A549s, again, 1,000-fold. Human PBMCs, 71 micromolar. THP1s, 100 micromolar. We're extending this across a bunch of we haven't found a cell other than a parasite that's sensitive to this stuff in a remarkable way. All right, here's a bit of the bugaboo. Oh, first off, solubility for this is phenomenal. So easily soluble, that sometimes kills small molecule screens as solubility issues. So we don't have trouble with solubility. Where we are challenged is in T1/2. And so we gave it IP. So Kurt, again, helped us out developing a mass spec assay. We've given it IP and we've given it orally, and we get almost the same result. massive absorption, either IP or orally, into the 10,000 or so nanomolar, way above what we need, but then a very rapid fall off. We can, if we give it IP, we can do QA dosing. This was at 5 milligrams per kilogram. But what we really want to do is flatten out the top of that curve. What I didn't know is when you give it IP, you're actually subjecting the drug to first pass. And it depends on how big the drug is, whether it gets absorbed on the parietal or on the visceral membrane. And so in our case, we're pretty sure we're getting first pass metabolism. So even as we speak, we're testing it out sub-Q. And I don't know if Kurt's on this one, but hopefully we'll have some samples for you soon, pal. Next couple of days, actually. And if we still don't flatten that curve out, we're going to look at some microencapsulation. And there are a bunch of folks at Brown, including in my department, who have expertise in that area. So that is drug 19. We're pretty excited. We think we can get the mouse model of infection to, we can dose this sufficiently that we should have activity there. And so that experiment we'll be doing after the holiday season. It's kind of a heavy lift for the team to do it. So take home messages, and I'll take some questions. Huge killing machine. It's going to continue to kill. It's getting worse now that we have drug-resistant parasites that are resistant to artemisinin in sub-Saharan Africa. Jeff Bailey in our department actually is really a world's expert in that, tracking it. It's going to get worse, no question. And so we're pretty excited. Yeah, we are going to move on to some hopefully phase 1 studies in humans with a vaccine, but I'd love to get this drug into a primate model, but that's $250,000. So huge number of folks. Patrick Duffy and Michal Fried for Patrick was my postdoctoral advisor for continuing to give me access to their field collected samples. Chris Nixon and Deepak did the cloning early on for GARP, and others. Whole pile of people have been involved in the killing assays, the imaging assays. Drew Weissman, of course, did the lipid encapsulated mRNA. That was back before he was famous in 2017. And 2 folks stand out in terms of their drug. 3 folks stand out in terms of their drug work. Adele Nefsi is our medicinal chemist. Andrew Lenikoff designed the inhibition of antibody binding assay and did the initial screen. And Tanbir and Nadrana in my lab, who's been doing almost all the drug assays, but with a lot of help from a bunch of other people. And of course, always remember your overseas colleagues who helped define the populations. They're absolutely critical, as well as the study subjects themselves. With that, I'm going to go through 3 slides and then take questions. So this is a CTR slide. Thanks for attending. These are the next 2 speakers, Molly and Dan. Dan, I know, will be talking about his more recent work in looking at sperm RNAs across males with various fertility issues. He'll give a great talk and some more save the dates. And with that, one last thing for any junior faculty or students that are on the list. Remember, the reason I show this picture, if the problem is big enough, it will take you a very, 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 very long time. And so be prepared to lose your youth on the thing you focus on. And if you choose wisely, you'll be thrilled to have spent your life working on the problem. And with that, I will take any questions.
Okay, great. Thank you so much, Jake. That is absolutely terrific story. So, number of questions. Spade wonders, was the goal of the substitution screen to find a molecule with a longer half-life? And if so, have you and Kurt looked to see what the metabolites are? And perhaps there's a specific site that you can modify to reduce the rate of metabolism.
Absolutely great question. So for the Cambridge screen, we were initially doing the SAR by catalog to find a more effective, i.e. lower concentration required for parasite killing. That was our initial plan. And the half-life for, let me step back for a second. What kind of drug do I want? This is going to sound crazy. If you look at the fundamental plank that supports everything we do with antibiotic stewardship, it is that we have a SWAT team in the pharmacy that will tackle and potentially kill any physician who prescribes gorillacillin for a trivial infection. And we all know who they are. You get attacked from behind, they trip you. They're like, are you out of your mind giving gorillacillin for a simple skin infection? So that is designed to limit the generation of resistance to our newest and best antibiotics. In malaria, in a village in an endemic region, in any of, literally a mud hut will have what's in Kenya, it's called a duka, which is a very simple shop, basically. They'll sell soap, they'll sell eggs, they'll sell cooking oil, and they'll sell artemisinin, our best antimalarial. The drug that we use for severe malaria is the exact same drug that's taken over the counter by mouth, all of the problems associated with. In malaria, we violate the prime directive. We do not reserve a class of antimalarials for severe malaria. So what I would dream about is a suppository, IV or sub-Q drug, suppository so you can get it at that first point of healthcare contact in a dispensary. Remember, the child is going to, severe malaria, these are very frequently very obtunded individuals. So suppository or sub-Q or IV. Suppository at the dispensary, sub-Q, IV at the, tertiary, not tertiary, but more advanced care facility. So the T1/2, and remember, with allometric scaling between mouse and human, that's going to go typically a 2 to 4-fold increase in T1/2. That, I think, for the 579 compounds would work. For the new compounds, the combinatorial library compounds, I think we've got to I'm learning the buzzwords. Oh, how did that happen? I'm learning the buzzwords of the drug developers. The buzzword is we need to harden the molecular soft spots. Like, what the hell does that mean? But I think it goes directly answering your question, Dan, is that our medicinal chemist is now looking at that drug 19 from the Cambridge combinatorial library to enhance its half-life. Kurt and his former postdoc Kate did unsupervised, I want to say the word metabolomics, but I don't know what they really call it, where they look for all the different metabolites. And she gave me this list of what she thought the cytochrome P450 enzymes were that would be metabolizing the drug. So we're trying to do that. I think it's going to be more important for the combinatorial chemistry library compounds, because those really do have half-life issues.
So another comment from Edith Messiahowitz is perhaps transdermal administration, which is an interesting thought too. So I have a question about GARP.
Yeah.
What is GARP? Is it a mitochondrial protein that's unique to this parasite?
No, I can't. So I have a hypothesis, but it'll sound like I'm a total nutty cuckoo guy. But I'll tell you the hypothesis. So GARP is no homology to anything. So I can't, by similarity, I can't tell you what it is. I truly don't know. What I can tell you is no parasite in their right mind wants to kill its host. It's a dead end. It reduces your reproductive fitness. The best parasites, the ones with the longest coevolution, and malaria is not a long coevolution, but the ones with the longest coevolution modulate themselves to their host. Bacteria have a thing called quorum sensing, and there are all kinds of flavors of it, where they know how many of their friends are there. And if there are too many friends, they shut down the replication rate. Trypanosomes know how many of their friends are there, and their method is fascinating. There is yet to be a density-dependent regulatory mechanism discovered for malaria. Here's what I think. You can have for me, if I get 1 or 2 parasites, I'm sick as stink. For a Kenyan kid with partial immunity, he or she could have thousands and thousands of parasites and still go to school. Now, they're still sick, don't get me wrong, but just sensing the number of parasites is not the most efficient system. Sensing host distress is the way to go. If I were the evil god designing this parasite, I would have it sense host distress. Specifically, I would have it sense an acute inflammatory protein. I actually teased that in our paper. So I'm on record before. We did a screen to try and find host ligands that bind GARP. It was done by Errol Fickrig, the chief of ID down at Yale. He's got a yeast surface display library of the entire human exportome, 6,000 human exported proteins. And he found several hits. My favorite, CCL5 binds GARP. And that binding is significant both by NMR and a version of SPR called mesoscale thermophoresis. So it really does bind. And when you add CCL5 to parasites in culture, they die. And 5 published papers, patients with severe malaria have paradoxically low levels of CCL5. It's an acute phase reactant. It's not a negative acute phase reactant. And so what I think is GARP is on the surface to sense host distress. Our antibodies and our small molecules are co-opting the regulatory mechanism for the parasite. That's what I think. I can't prove it yet. We have a bunch of data, but there are 1 or 2 flies in the ointment, unfortunately.
Okay. And then another quick question from Jerry now. Are there compensatory mutations in the GARP knockout parasite that could suggest reduced reliance on GARP if therapy or vaccine are deployed? In other words, resistance.
Yeah, it's a great question. Let me share my screen again. How do I do that?
Really fast.
In that case, I'll just tell you. So a GARP knockout parasite is a more rigid red blood cell, and it can't make it through the cords of Billroth. So I don't think a GARP knockout will be viable.
Okay. Very good. Well, thank you, Jake. What a fascinating story. And
my pleasure.
The thing I love is that a pathologist has become a pharmacologist. How did
the pharmacologists hate it though? They won't let me in the room.
But that's really, but the journey is a really great story, and hopefully soon to be saving lives. And
thanks so much, everybody. Really appreciate all the comments. I will definitely read through them. There's some great ones here.
And we'll see everybody, I hope, next month for our next seminar series, which will be featuring 2 of our Advance-CTR awardees. So bravo and have a great day.
Take care all.